Okay, so last time we ended up here, right, with the auto brewery syndrome, which also had an East Asian link there with alcohol dehydrogenase deficiency. Yeah.
I saw the article in my the other day.
Ah, okay. So now lactic acid and ethanolic fermentations are very simple fermentation pathways with just one or two enzymatic steps between pyruvate and the fermentation product. So many bacteria can use these. Well, there are a lot of lactic acid bacteria, but there are also more complex fermentation pathways, which we're going to look at now. So one of them is butanediol fermentation. So you start off with pyruvate. And what's going to happen here is, you remember pyruvate's 3-carbon molecule. But the first step is to combine two of them together. That gives you acetolactate. So that's 5 carbons. And it kicks out one molecule of carbon dioxide. Then once again, another molecule of carbon dioxide comes out. This gives you acetoin. And then this will be reduced by NADH to recycle the NAD plus to give you 2,3-butanediol. Now, this is very common in gram-negative bacteria. A lot of them do that. So gram-negatives, several genera of gram-negatives and some gram-positives. One of the things I kind of wonder about this is why are they doing something? This is 3 enzymatic steps, not just 1 or 2. It's more complicated. Why do they bother to do something more complicated? Well, one of the reasons is perhaps that the end product is a little bit less toxic than lactic acid or acetate. The butanediol won't have an effect on the pH, and is probably a little bit less toxic than ethanol. So it's worth doing this. Industrially, though, this used to be an important industrial process because butanediol was a starting compound for the synthesis of artificial rubber. But it's generally been replaced by petrochemicals. However, there's some kind of there's some renewed interest in using this fermentation process to produce butanediol for use in petrochemicals, specifically from glycerol, because glycerol is apparently a side product from the production of biodiesel, or perhaps more accurately should be called agri-diesel, okay, from palm oil or something like that. So this biodiesel or agri-diesel production produces a lot of glycerol as a byproduct. So people are starting to think, well, can we use this glycerol for something? And so there are people who are trying and find the best bacteria that can ferment glycerol and produce butanediol, and try and work on this to get good high yields that can be exploited industrially. So this is a paper published just last year with the the glycerol concentration going down and the butanediol concentration coming up, okay, over the fermentation time, by Klebsiella oxytoxa. So that's still kind of useful. Now, the last 2 types of fermentation are going to be a bit more complicated. And in order to understand them, I think you need to have an idea of what pyruvate can also be used for in bacterial metabolism. So when bacteria are not performing fermentation, pyruvate is still an important molecule because can be carboxylated to oxaloacetate, and this is an important precursor for the biosynthesis of amino acids, and it's also something that will lead into the Krebs cycle. So the Krebs cycle, this is an essential part of respiration. So facultative anaerobes that can metabolize glucose by respiration and also by fermentation, they will have the Krebs cycle, okay? And the Krebs cycle, well, it starts off with the combination of acetyl coenzyme A and oxaloacetate to give you citric acid, and then this citric acid will cycle around to oxaloacetate. So another, use of pyruvate is to produce acetyl coenzyme A that feeds into the Krebs cycle. And this reaction is catalyzed by pyruvate dehydrogenase, which will once again produce one extra molecule of NADH. So during respiration, that's not a problem because this NADH will be oxidized, well, finally by molecular oxygen. So all you have to remember is, okay, in bacteria that are performing respiration and fermentation, they're going to have these pathways around, okay? Now, propionic acid fermentation looks complicated, okay? You start from pyruvate, gets carboxylated to oxaloacetate, and then this will be reduced through to succinate, and then one final molecule of carbon dioxide is kicked out and you get propionic acid. So once again, this looks like overly complicated. You've got 1, 2, 3, 4, 5 enzymatic steps from pyruvate. You think, why are they doing something so complicated? Well, when you look at it, in fact, all of these are just Krebs cycle enzymes running in reverse. So if you've got a facultative anaerobe that will use respiration when there's still some oxygen around, and then will switch to fermentation when the oxygen runs out, then it kind of makes sense because they already have all these enzymes hanging around in the cytoplasm of the cell. So you've just got to start expressing one extra one here, and then you can switch respiration to fermentation. It's very easy. So in fact, it's simpler than it looks. Now the last one is mixed acid fermentation, which is very, very common in a lot of gram-negative enterobacteria. Now once again, these are facultative anaerobes, these bacteria, so they can perform respiration when oxygen is present. Okay, so once again, with mixed acid fermentation, it all starts from pyruvate. The first step is to produce acetyl coenzyme A. But the enzyme that does this is not pyruvate, what is it, pyruvate dehydrogenase? Yes, I believe so. Just go back a little bit. What did I say here? Yeah, pyruvate dehydrogenase. So it's not that, because pyruvate dehydrogenase will produce one extra NADH. And you don't want that if you are fermenting, because the whole point of fermentation is to recycle the NADH into NAD+. So for mixed acid fermentation, the step from pyruvate to acetyl coenzyme A is catalyzed by this enzyme, which is called pyruvate formate lyase, and will produce formic acid and acetyl coenzyme A. And then from here, from acetyl coenzyme A, then there are a bunch of different branches that can be used. Some of them will recycle NADH into NAD+. So that's the main point of fermentation. But other ones can be used to produce some ATP. And this, I think, well, at least that's what makes sense to me, because every time I see something complicated in biology, I always think, well, why is it so complicated? And there's always got to be a reason. And I think the reason here is if you perform a mixed acid fermentation, then you can squeeze out 1 or 2 extra molecules of ATP from the process. And that makes sense because the whole problem with fermentation is that it doesn't produce enough ATP compared to NADH. So if you can do something clever, then you can produce an extra bit of ATP. Now, the big problem with this is the buildup of acid fermentation products, because for every molecule of pyruvate that gets goes through this fermentation pathway, you produce one molecule of formic acid and a bit more of some of these other acidic products. And when Escherichia coli is growing at pH 8, then for every 100 micromoles of glucose, there'll be 50 micromoles, or even, no, 86 micromoles of formic acid, which are produced. Now, that's a problem because formic acid will acidify the cell culture medium. So once the pH goes down, then bacteria E. coli will start producing this other enzyme, which is called, I think it's called formate hydrogen lyase or something like that. So it's going to split the formic acid into molecular hydrogen and carbon dioxide. So once the pH goes down, then you get more and more carbon dioxide and hydrogen gas produced and less of this formic acid. And then these can be used by methanogens. Okay, so this is where the source comes from the production of methane in the guts of ruminant bacteria, ruminants, okay? So it's this mixed acid fermentation which will give as fermentation products a mixture of hydrogen and CO2, and these can be used by methanogens. Okay, so one last thing on fermentation. So we finished with sugar fermentation, but carbohydrates and sugars are not the organic substrate, not the only organic substrates that can be fermented. So some bacteria can ferment amino acids, notably bacteria of the genus Clostridium. And this is illustrated by the fermentation pathway here. So this is what's also called the Stickland reaction. Now you don't have to remember any of the details here. You just have to realize that the two substrates are what? Alanine and glycine, two amino acids. And you have one of them, which is going to be the electron donor, the other is going to be the electron acceptor. So at the end of this process, you get, what products do you get? You get carbon dioxide, you get ammonium, and you get acetic acid. And somehow through this fermentation pathway, you can get some ATP being produced. Now, this is particularly a problem in tinned food if the sterilization process hasn't been done correctly, because Clostridia, which are spore-forming bacteria, they can survive heating at 100 degrees. So if you just boil them, in your pan at home, then you'll kill vegetative bacteria, but endospores will not be destroyed. And then later on, if you have any spores from Clostridia inside your tin can, they can germinate because they're present in a rich food source and then start performing this Stickland reaction. And this will produce gas, CO2, so that's what causes the deformation of the cans. So if you have any old tin cans in your house and you see them like this, they're buckled, there's been gas produced in the inside, you should never eat anything that is in the tin can like this, okay? Because one of the other problems is that the genus Clostridium also contains this pathogen, Clostridium botulinum, which produces botulin, a very powerful exotoxin. And there was a case just last autumn, of people being poisoned in Bordeaux. If you read this here, we had 12 people who were intoxicated and one woman was killed after eating these sardines that were, home conserved sardines that the bar owner had made and then sold. So unfortunately, they've been contaminated with Clostridium botulinum. They grew, even though the container was closed, and then some of the people ate them, and they got sick, and one person even died. Okay, so see what you remember about fermentation. Okay, what do we got? Okay, so the top two answers are correct. One of the drawbacks is end products can be toxic. Acidic fermentation products will inhibit bacterial growth, even if there's still plenty more sugar around. The pH goes down too far, many bacteria cannot grow anymore. The other main problem is, number one, energy yield per carbon is low, like ATP yield per carbon is very low. So 1 or 2 molecules of ATP per molecule of glucose. And the other three answers are false. Number 3, no, gram positives and gram negatives. Plenty of gram negative bacteria perform fermentation. And 2 is wrong. It's the other way around, okay? Fermentation, there is no external electron acceptor, oxygen or anything else. So that's very important to remember. Okay, so that's it. That brings me into the summary here. So what are the problems with fermentation? That there's too much NADH compared to ATP, and it has to be, the NADH has got to be recycled to NAD+. For this, there needs to be some kind of electron acceptor, but it's an organic metabolite, pyruvate, something derived from the original carbon source is the electron acceptor. Lots of nutritional food industry, industrial uses of fermentation products, and it will occur either when there is no oxygen or when there's a lot of sugar around. Okay, so in the lab class, and I don't know if you remember when you saw, well, when I showed you what a plate of Chapman agar looks like with Staphylococcus aureus. So when Staphylococcus aureus colonies grow on the agar, on Chapman agar, they will ferment the mannitol and reduce the pH of the agar, giving a yellow color. But the bacterial colonies, they're on the surface of the agar. There's plenty of oxygen around, but they still ferment because there's so much sugar around. They don't care, Yeah, look, 1, 2 molecules of ATP, that's fine. I've got tons of mannitol. Let's just eat it all up, ferment that sucker. Okay, so when there's no oxygen or when there is really a lot of sugar, then fermentation can occur. Okay, so I start with fermentation because, well, unfortunately, it's probably the simplest way to get energy. and all the others are more complicated. So among the complicated ways to generate metabolic energy, let's start with respiration. Now, have you seen this before in biochemistry? The general steps of respiration. Who says yes? One person. Who says no? I don't know anything about this. Most people don't know whether you've seen it or whether you or haven't seen it. Oh, knowing that pachinko is Korean, that's okay. Whether I've seen something about respiration in any of my lectures previously, oh man, too difficult, that question. Please ask me another question about Korea. Okay, so let's just imagine that you don't know so much about this. Okay, glycolysis, we've seen that. You start off with some kind of glucose and you metabolize it to pyruvate, you get some ATP and some NADH. Then if oxygen is present and you have the metabolic capacity, then you can carry on and use respiration. So what is going to happen is that this pyruvate is going to feed into the Krebs cycle. It's going to be completely oxidized to carbon dioxide. So when the carbon is being oxidized, something else has got to get reduced, and this is NAD+. It will be reduced to NADH. So you get a whole ton of NADH in the Krebs cycle. And then the last step is electron transport and oxidative phosphorylation. So electron transport is going to take the electrons that are currently on all the NADH molecules that have been produced, and the electrons are going to be pushed onto an external inorganic electron acceptor, which is oxygen, right? Aerobic respiration, oxygen. So that's good, that works. And during this process, then bacteria and our mitochondria generate a proton motive force, and this proton motive force is what's used to generate ATP. So in Escherichia coli, the electron transport chain is simple. It's got three components. Okay. Now, I'm not going to talk about the Krebs cycle. That can be a special treat that you can save yourselves for biochemistry classes. So imagine, we've done the Krebs cycle. All the carbon in the glucose is oxidized to CO2. We've generated a huge amount of NADH. So we start here with NADH. So the first thing is NADH dehydrogenase takes off these electrons, transports it onto quinone. I'm telling you, this is so annoying. I have no idea why these. Oh. Okay, that's better. I have no idea why those were missing. So the electrons are transported from NADH onto quinone. Quinone is what? Quinone is this molecule. It's a hydrophobic organic molecule. So this always stays dissolved in the plasma membrane. And the part that is, the business end of this molecule is here, is this head group. So this is the oxidized form of quinone, which is called Q here. And the reduced form will transform these two kind of ketone groups to hydroxyl groups. So this becomes OH, this becomes OH. And two electrons will be transported onto from Q to the reduced form QH2. So the reduced form then migrates through the membrane and goes and interacts with the cytochrome oxidase complex. And the cytochrome oxidase complex is going to pick up the electrons from the reduced quinone, the QH2, but the protons that were picked up in the cytosolic phase are liberated on the periplasmic phase. So just this quinone cycle generates some of the proton gradient across the membrane. You pick protons up here and you release them on the periplasmic face. Then the electrons from the cytochrome oxidase complex, so cytochromes are what? They are these small proteins with a heme group and an iron atom here, which is coordinated, and this is what's going to be doing the electron transport. So the cytochromes are finally going to transport the electron onto an oxygen molecule to produce water. And this is going to use up protons on the inside of the bacterium. So the overall result of this is that protons are used up on the cytosolic face of the plasma membrane and they are liberated on the periplasmic face. So you get a proton gradient across the membrane. Now E. coli has got two cytochrome oxidase complexes, the BD branch, which is used when oxygen is in low supply, so it's got a higher affinity for oxygen, and the BO branch has a lower affinity, it only works if there's a lot of oxygen present, and what is good about it is that it can also translocate two extra protons across the membrane, so it's more efficient at generating a proton gradient. So that's electron transport, and the last step is production of ATP. So this is the proton-dependent ATPase, which can phosphorylate adenosine diphosphate to ATP by using the energy from the transport of 3 protons across the plasma membrane. So if you add up all the molecules of NADH that are produced by oxidizing glucose to CO2, and then count how many protons are translocated across the membrane by the electron transport chain, and divide that by 3, well, then you end up with a maximal theoretical yield of respiration of 38 molecules of ATP per glucose. So that's 10, 20, 30 times more efficient at producing ATP than fermentation. So it's really, you're really getting way more ATP out of this. And that's the advantage of aerobic respiration. That's why it's worth the risk of bacteria performing this risky metabolic step, which is to add electrons onto a molecule of dioxygen. As we saw with the oxygen relationships, when you push electrons onto a molecule of oxygen, you can produce oxygen radicals, which are very, very reactive and toxic. And that's why aerobes and facultative anaerobes must express superoxide dismutase and catalase. Anyway, so it's worth taking the risk of playing around with toxic oxygen molecules because you get a big ATP payoff. Now, there are also bacteria which can perform anaerobic respiration. Now, I want you to just think about this term for 1 second. Anaerobic respiration, because on the surface of it, it does not make sense, right? Respiration, you breathe, you need air for it, obviously. But here we mean respiration is in the biochemical term. That is, something is being used as an external electron acceptor. And here, bacteria can use nitrate as the electron acceptor. Why does this work? Because the whole thing that is happening here is you've got NADH is the electron donor, and the electrons have got to be pushed onto an electron acceptor. So if you've got oxygen down here, oxygen is the best electron acceptor, so it's easy. It's a very energetically favorable reaction. But there are other good electron acceptors like nitrate and nitrite are pretty good electron acceptors. So if all the oxygen has been used up and there's still some nitrate, then some bacteria can use the nitrate as the terminal electron acceptor in respiration. So the first steps are the same: glycolysis and the Krebs cycle, and that gives you a lot of NADH. The last step, the electron transport, is different. So this is the aerobic electron transport chain in E. coli with NADH at the start and molecular oxygen at the end. So here is an electron transport chain for anaerobic respiration in the presence of nitrate. So it begins with the same kind of thing, the NADH dehydrogenase and the quinine cycle, Q cycle, quinone cycle rather, not quinine, but it ends up with electron transfer onto nitrate and to nitrite. And the end result is similar. You get this proton gradient. The proton gradient is what allows the bacterium to produce ATP. So anaerobic respiration works with nitrate as an electron acceptor, but can also work with other reasonably good electron acceptors. So in the water table, for example, you'll have a kind of stratification with different bacteria growing depending, so you have to imagine where's the energy coming from to begin with. It's coming from organic carbon. And the organic carbon is going to be metabolized first by respiration, as long as there's some oxygen around, okay? Then at some point, all the oxygen is going to be depleted. Then what's going to happen? Then some bacteria will switch over to anaerobic respiration, using first nitrate, then manganese if there's any around, then iron if there is any, then sulfate, and then finally CO2 as the electron acceptor. So different types of bacteria can use different electron acceptors. So enteric bacteria can use nitrate, desulfur vibrio, desulfotomachia can use sulfate as an electron acceptor. Then you use up all these things. They use one after the other for the best electron acceptors first, and then the worst are used last. And then finally, when there's nothing left, well, then some bacteria can still ferment if there is any organic carbon left. Okay, so just keep that in mind. So that's respiration and anaerobic respiration. Now the next thing, which is kind of similar, is chemolithoautotrophy. So chemolithoautotrophs generate their energy from a favorable inorganic redox reaction. And there are basically three types: hydrogen oxidation, nitrogen oxidation, and sulfur oxidation. So hydrogen oxidation is a bit, exceptional, but these two, nitrogen or ammonium oxidation and sulfur oxidation, this is just like anaerobic respiration in reverse. And the reason that this can work and bacteria can get energy out of the process is that the electron transfer, the redox reaction, can be energetically favorable if there is a better electron acceptor present. Okay, so if you've got a reduced form of nitrogen, like nitrite, and you want to use that as an electron donor, can only work if you've got an even better electron acceptor present, like molecular oxygen. Okay, so this reaction, oxidizing nitrite to nitrate, can be used to generate metabolic energy if molecular oxygen is present. Okay, so I hope that's kind of how it's working. So there's some kind of favorable redox reaction. And based on this, all chemolithoautotrophs have got to be able to generate the three forms of biochemical energy: ATP, proton motive force, and NADH. Now the easiest thing, the way to do this is if you've got molecular hydrogen available. So there are hydrogen oxidizers which are aerobic. They just oxidize molecular hydrogen with molecular oxygen and it produces water. So these names are kind of easy to remember. Hydrogenophaga, it eats hydrogen. Aquifex, it makes water. And then there are also inorganic hydrogen oxidizers, which are the methanogens, like Methanococcus. So these hydrogen oxidizers can actually use carbon dioxide as the electron acceptor and get methane out of it. So this is, the absolute worst electron acceptor, but it can only work if you've got a really good electron donor to provide the energy for the process. Okay, so hydrogen oxidizers kind of have it easy because hydrogen is such a good electron donor that these bacteria can produce NADH just directly, because this is the standard redox potential of this half reaction. The more negative this value is, the better the electron donor is. So if you put these two reactions together, it will work, okay? So these hydrogen oxidizers, they can generate NADH, and then they can have an electron transport chain, generate a proton motive force, and generate ATP. Easy. Okay, they can just do it. Especially when oxygen is present. Okay, methanogens is more difficult. And basically only archaea can do this. Okay, so that kind of makes sense. Now, how about sulfur oxidizers? So these are bacteria which will make a living by oxidizing reduced forms of sulfur like hydrogen sulfide or molecular sulfur, which you can find in some mines. And once again, there are some sulfur oxidizers which are aerobic, they require molecular oxygen, and others which are anaerobic. So aerobic sulfur oxidizers like Thiobacillus ferrooxidans, basically they are getting reduced forms of sulfur like sulfide and iron. They are oxidizing them with molecular oxygen, and this will produce sulfuric acid and ferric iron. Now this is a big problem in abandoned mines for which had iron ore, because these bacteria can grow on the open mine seams where you've got iron ore present and sulfide. And this is a big problem, can cause thousands of tons of sulfuric acid to run off into the rivers that are draining these abandoned mines. So fortunately, the answer is very simple. You've just got to block off the opening to the mine, cut off the oxygen supplied to these bacteria, and then they can't produce sulfuric acid anymore. Thiobacillus denitrificans, this is kind of clever. It's got reduced sulfur, and the electron acceptor is nitrate. And this is still favorable because on my kind of redox scale, the redox couple between sulfide and thiosulfate is about here, it's about 0 volts. So hydrogen sulfide is a better electron donor than nitrite, for example. Okay, so in physical chemical terms it works, but sulfur oxidizers also have a kind of biochemical trick to generate ATP, which is that sulfur or sulfite can be oxidized to sulfate, or sulfite can be integrated into ammonium persulfate by combining with, adenosine persulfate by combining with adenosine monophosphate. And then these adenosine persulfate molecules can then react with inorganic phosphate and gives you 2 molecules of ADP. And then combining them to give you 1 molecule of AMP and 1 molecule of ATP gives you some ATP produced directly. And then, of course, the electrons, they go off into the electron transport chain, are pushed off onto the electron acceptor and then can generate a proton motive force. Okay, how do we get NADH? I'll show you that in the next couple of slides. Now, the next chemolithoautotroph are nitrogen oxidizers. For example, nitrosomonas will oxidize ammonium to nitrite, and nitrobacter will oxidize nitrite to nitrate, and they both require the presence of molecular oxygen. So generally speaking, this can still work because oxygen, molecular oxygen, is the best electron acceptor. It can work in physical chemical terms. But in terms of generating useful biochemical energy, it's difficult because the nitrite is a very poor electron donor. So the electrons that are on the nitrite here, they can't really be transferred directly onto NAD+, because this is a very, very good electron donor and nitrite is a very poor electron donor. So how can these bacteria, so they can't produce NADH directly. So how do they do it? Okay, how can they do this? Well, what they manage to do is, okay, so they can't get NADH directly, but what can they do? They can generate a proton motive force, because these electrons here, they're going to go onto oxygen at the end, basically. And what nitrifying bacteria have is a couple of enzymes that are very interesting for biochemists: ammonium oxidase and hydroxylamine oxidase, HAO. So what these enzymes manage to do is produce nitrite from ammonium and strip off the electrons that are fed into this electron transport chain. And the end result is the export of protons. Electron transport chain, they go down the electron transport chain, and in the end, it's molecular oxygen that is the electron acceptor. So this generates proton motive force. And the proton motive force can be used for two things. One of them, generate ATP with the proton-dependent ATPase, and also it can force the NADH dehydrogenase to work the other way around. Okay, so this is what's called reverse electron transport. If you've got a strong enough proton motive force, then you can force NAD+ to accept some extra electrons and generate NADH that way. Okay, so, chemolithoautotrophs, they're doing difficult things, but they manage to generate ATP, proton motive force, and NADH from some inorganic chemical reactions. Okay, so what I want you to what I want is to see if you can understand why this works, okay? Just in a physical chemical sense. So I don't know if this is going to work with a kind of clicker question, but I thought maybe I'll just put them up here and I'll try and explain to you the reasoning and see if you can figure out the other ones, okay? So what I've got here is some kind of proposed bases of metabolism. One of them is going to be the electron donor, the other is the electron acceptor. Can it work? And what you have to do is to look up on the redox scale here. The ones at the top, these are the best electron donors. As you go down, they're poor electron donors, they get to be better electron acceptors. So what you have to figure out is something that is going to be energetically favorable is the electrons go from here to somewhere lower. So my first proposition is glucose is the donor, nitrate is the acceptor. So glucose getting oxidized to CO2, really good electron donor. Nitrate, the couple is here, so yeah, that's going to work, right? Very good electron donor, very good electron acceptor. It can work. And this is anaerobic respiration. Okay, what about this one? Everybody thinking about this? Everybody ready to express an opinion? Who says yes, it can work? Some people. Who say no, it can't work? More people. Well, it's really borderline. It's really, really tight because you're just going between like here and here. It's like a tiny, tiny difference in tiny potential difference. So maybe yes, but it's really, really difficult. Okay, but this is what's basically happening. What methanogens are doing. Okay, they're just scraping a living off a very, very marginal redox reaction. How about this one? Nitrite as a donor, CO2 as an acceptor, and it produces acetate plus nitrate. I think I'll draw this one up on the board. So what am I saying here? I'm saying that we've got at the same time don't worry about where the carbon is coming from. Okay, who says this is going to work? Nobody. Who says it's not going to work? Yeah, you're right. It can't work, right? What about the opposite? Can that work? Could you oxidize acetate into CO2 with nitrate as the acceptor? Yeah, that would work, right? So basically, this one cannot work in that direction, but it can work this way. Okay, this could work. Okay, how about this one? Ammonium as the donor, thiosulfate as the acceptor. And for this one, I put the redox couple with thiosulfate to sulfide on here. Everybody thought about this? Okay, who says, yes, it can work? Who says, no, it can't work? Not many people putting their hands up. Okay, so let me draw this one again. Ammonium as the donor. There's no ammonium on here, okay? That's the problem. Okay, let's say nitrite as the donor again, okay? Because it's the same kind of thing. So I'm saying this is going to work, and we have thiosulfate as the acceptor, thiosulfate, yeah, and nitrite as the donor. Would that work? No. But the other way around it works, right? Yeah, exactly. Okay, so that's how this works, this stuff. Hydrogen sulfide as donor, nitrate as acceptor. Who says yes? Oh yeah, it's the other way around, right? Basically, it's the other, this is the, okay? The other way around it works. And can everybody see this or not? Oh man. I'm guessing that you don't really get this. Okay? So what is happening here? Hydrogen sulfide as a donor. Hydrogen sulfide is up here. Nitrate is the acceptor. Nitrate is down here. So yeah, it will work. This is favorable. It's the opposite of this one, basically. This way around, it can't work. This way will work. Okay? So the kind of bottom line of this story is, the underlying chemical reaction has got to be exergonic, favorable, okay, energetically favorable. So, and a lot of it depends on, whether molecular oxygen is present, whether a particular sulfur or nitrogen compound will be functioning as an electron acceptor or as an electron donor, okay? Okay, so we have, we're going to be finishing on time, I think, which is good. So what's the last thing we have to talk about? That's photosynthesis. Now, there are many types of photosynthetic bacteria. So some are archaeobacteria, but most are eubacteria. And the pigment that is absorbing the light is different in eubacteria and in archaeobacteria. So halobacterium performs photosynthesis based on a protein called bacteriorhodopsin. Whereas all the other photosynthetic bacteria perform photosynthesis basically by the absorption of photons by bacteria chlorophyll. So what does this mean? This means that photosynthesis was invented or evolved in bacteria twice. Once in archaeobacteria with a system based on bacteriorhodopsin, and once in eubacteria with a system based on bacteria chlorophyll. Okay, just two origins. But when you look at the different types of photosynthetic bacteria amongst eubacteria on 16S ribosomal RNA phylogenetic tree, you can see that the photosynthetic bacteria, they're kind of all over the place. Okay, so all of these different groups of photosynthetic bacteria did not inherit the photosynthetic system from a common ancestor. So there was one original photosynthetic bacterium amongst all of these guys, and then some transfer of genes from the photosynthetic bacterium to others to produce this kind of like a polyphyletic character of photosynthesis among bacteria. Okay, so how do these photosynthetic systems work? So bacteriorhodopsin, which is in Halobacterium salinarum. It's a 7 transmembrane helix protein, which has a pigment kind of on the inside of the pore here, across the membrane. And it functions as a proton pump. So this is the cytoplasmic phase, and this is the periplasmic phase. And when light is absorbed here, it allows the bacterium to translocate one proton across the plasma membrane. So basically, this is a kind of light-activated proton pump, and it allows the bacterium to generate proton motive force. And if you've got proton motive force, you can generate ATP. So it's good. No electron donor, no electron acceptor. Okay, so getting back to that's the only photosynthetic system we know in archaeobacteria. Okay, so some of them have this bacteriorhodopsin, it does that. Now, what about the photosynthetic eubacteria? So I checked up to make sure this was still the current model. So there seems to be a consensus that the bacteria that first developed photosynthesis were the ancestors of the cyanobacteria. So what is proposed is that this kind of precursor to cyanobacteria had both photosystem 1 and photosystem 2. And then there were gene transfer events from this kind of ancestral bacterium to different groups of the currently photosynthetic bacteria. And these groups are quite ancient. This occurred a long time ago. So it's thought that these gene transfer events occurred before the origin of oxygenic photosynthesis, about 2.5 billion years ago. Now, cyanobacteria, they are the only group that contain photosystem 1 and photosystem 2 like plants. Okay? All the other photosynthetic bacteria have either photosystem 1 or photosystem 2, but not both of them at the same time. And that's kind of strange because if we understand photosynthesis in plants, we realize, oh, it's great, it allows plants to grow with just like nothing, just water and CO2 and light, and they can generate energy and all their organic carbon from that. But if you've only got half a photosynthetic system, why is that going to be useful for bacteria? Okay, so I just want to show two examples. So in chlorobene, the green sulfur bacteria, they only have photosystem 1. So what is happening here is that the photosystem 1 will, where's my arrow? Go get it, will absorb 1 photon. The electrons in the chlorophyll molecule will get all excited, and then they'll start the electron transfer down through an iron sulfur protein, and we'll be able to reduce NAD plus to NADH. And so there is an electron acceptor and there is an electron donor. The electron acceptor is NAD plus, and the electron donors are reduced sulfur compounds like hydrogen sulfide or thiosulfate. So why is this useful for the bacteria? This is useful because if you remember, in sulfur oxidizing bacteria, the redox potential of the sulfur oxidation is not enough to transfer the electrons onto NADH. So those bacteria have got to use some of the proton motive force that they generate to force the electrons onto NADH. Now, if you've got this kind of sulfur oxidizing metabolism and you add a photosystem 1 onto it, then the whole of your metabolism becomes more efficient because you can have direct reduction of NAD plus to NADH, and then all the proton motive force you generate can be used to produce ATP. So it's an advantage, okay? Now, what about if you've just got photosystem 2 on its own? So this is the case with purple non-sulfur bacteria. So in purple non-sulfur bacteria like Rhodobacter, then what is happening is that the electron in the reaction center gets activated, and then it gets transferred onto quinone, and then onto cytochromes, and then it goes back down and goes to the reaction center. So the electrons just go round and round and round and round. Okay, there's no electron donor, no electron acceptor. Why is this useful? Well, it's useful because if you've got the Q cycle, the quinone cycle, you can produce a proton motive force. And you can use this proton motive force to generate ATP. So that's great. You get ATP for free. So that's why it's useful. So Rhodobacter, purple non-sulfur bacteria, they are basically chemoheterotrophs. They require, they're basically, photoorganoheterotrophs, should I say. So they require organic carbon as a source of electrons and a source of carbon for making their organic structures, but they get energy from light. And these bacteria can work in both a kind of photoorganoheterotrophic way, or if you culture them in the presence of a nice amount of glucose, culture them in the dark, they can probably still grow as chemo-organoheterotrophs. But their metabolism is more efficient if light is present, because then they can get more energy, because they're anaerobes as well. They don't perform respiration. So as you remember, if these guys are living by fermentation, they won't be getting much ATP per organic carbon molecule. So if they can add a bit of photosynthesis here, then that means they can produce more ATP, and it helps their metabolism become more efficient. Now, the last kind of photosynthetic bacteria are, as you know, cyanobacteria. So these are basically just the same things as chloroplasts in plants, and they are both photosystem 1 and photosystem 2. So in this case, what is kind of amazing thing about this is that combining these two photosystems allows water to be used as an electron donor. And this is the most unfavorable chemical reaction probably like in the world, okay, in the whole of biology anyway. Well, it's right up there with nitrogen fixation, let's say. So it's a very, very unfavorable chemical reaction because, as we know, molecular oxygen is the best electron acceptor. So that means that water is totally the worst electron donor. And it's this kind of 4-manganese calcium reaction center that does the job here of stripping off the electrons of water molecules. But in terms of generating metabolic energy, what does this allow the bacterium to do? Okay, so you've got photosystem 2, which has this quinone cycle, generates proton motive force, you can get ATP. And then at the end, the electrons that are stripped off the water here can be transferred onto NADP+, so you get NADH. So you get all three things. You get proton motive force, ATP, and NADH equivalents, just with light from light and a universal electron donor, which is water. Okay, so this is a summary of the different types of photosynthetic metabolisms that exist in bacteria. So you have cyanobacteria with photosystem 1 plus photosystem 2. So these are the only oxygenic photosynthetic bacteria. So they are 100% photolithoautotrophs. Similarly, the sulfur bacteria, the green and the purple sulfur bacteria, they are photolithoautotrophs. Whereas the green and the purple non-sulfur bacteria, these are photoheterotrophs. So, yeah. So they get their energy source from light. However, their source from electrons and carbon for their metabolism must be organic carbon. So what has basically been happening here is that, these are the guys who've been like developing photosynthesis like from scratch, and then bits of their photosynthetic apparatus have been transferred out to other types of bacteria, which were basically either chemoheterotrophs or chemoautotrophs. And adding on a bit of photosynthesis to these groups of bacteria gives them a bit of an advantage. Now, as for the origin of photosynthesis in cyanobacteria, I think the prevailing view is that initially these bacteria were kind of like the green sulfur bacteria, basically using photosynthesis with sulfur, reduced sulfur compounds as the electron donors. And somehow they managed to acquire the ability to use water. But because these events happened, more than 2 billion years ago, it's very, very difficult to get any clear indications of the different steps in that process. Okay, so that brings us to the end of bacterial metabolism and the first 5 lectures. So, okay, the way I look at it anyway is that Okay, respiration. What is happening here? All the energy comes from a source of organic carbon to begin with. Then this is fully oxidized to CO2. That will give you a lot of NADH. Then these reduced NADH molecules will give up their electrons to an electron transport chain, which transfers the electrons onto oxygen or onto another good electron acceptor. This generates proton motive force, and you can get ATP. What is respiration for? Well, it's useful because you get a much higher yield of ATP than you can get from fermentation. And anaerobic respiration is not the same thing as fermentation, because in anaerobic respiration, you have the Krebs cycle, you have the electron transport chain. It's just the terminal electron acceptor that's different. It's not oxygen, it's nitrate, or it can be thiosulfate, or it can be, nitrate or thiosulfate mostly. Chemolithooautotrophs, well, they're kind of doing the opposite thing. The source of energy comes from some inorganic reaction, an inorganic redox reaction. So this oxidation reduction will allow the bacteria to start generating some energy by electron transport. And this electron transport will generate at least proton motive force. And then the proton motive force can be used to generate ATP and NADH. So that's how nitrogen oxidizers can generate metabolic energy. And then the NADH is used to reduce carbon dioxide into organic carbon to fix CO2. So instead of having organic carbon as the source of the energy in the beginning of all the metabolism, it's an inorganic reaction. It's the start of the metabolism, and the end product is organic carbon. And the tricky thing here, as we saw, is to kind of understand which donor-acceptor couples can work. And very often depends on, if you've got molecular oxygen present or not. And then finally, for phototrophs, it's understanding the biochemistry of photosynthesis takes a few lectures in plant sciences anyway, so we're not aiming to get the same level of understanding here. But you need to just understand that, there are different types of photosynthetic bacteria. They seem to have acquired photosystem 1 or photosystem 2 from the precursors of cyanobacteria, and that this has kind of been added onto some pre-existing metabolism that these bacteria already have or already had. Okay, so surprisingly, I'm about 8 minutes early, but I guess I will stop there. So the last microbe of the week, because the next lecture will be in about a month's time or more, that will be my last one. So this will be the last microbe of the week. When do we want the question to go out? Huh? This evening? Okay. This evening, 7 p.m.? Yeah, I can send that one out. Okay. I think it's going to be difficult.